if the tech will keep up.
There we go.
Fantastic.
Thanks very much, Thomas. And thank you very much for inviting me along to talk today. My name is Patrick Lewis. I'm a researcher here at the School of Pharmacy, and I've been working on Parkinson's disease for over 10 years now in a variety of different locations and doing a variety of different research. The task that Tom has given me is to give you almost a sort of potted history of Parkinson's disease and the major milestones that have really changed the way in which we think about the disease and help to develop some of the symptomatic treatments that we already have, and hopefully are laying the groundwork for research in the future which will help us to develop new drugs. And it's really exciting seeing the efforts of Parkinson's UK in really trying to drive that research. I've been funded by Parkinson's UK for the last 5 years, and a lot of the research that I do would not have been possible without the help and support of Parkinson's UK. So the journey I'm going to take you on today goes from 1817 over here in the left-hand part of the slide to the present day. So we've had 200 years since the first description of the disease that we now know as Parkinson's. And I suppose the best place to start is back in 1817 and with James Parkinson. And this is the frontispiece of his essay on the shaking palsy. And this really laid the foundations for our modern understanding of Parkinson's disease. There are actually versions of this available free on the internet. If you search for Essay on the Shaking Palsy, you can download a PDF of his original pamphlet on this. And I would really encourage you to read it. It's fantastically well written and actually incredibly insightful. A lot of, if you were to talk to a neurologist or a Parkinson specialist now, a lot of what James Parkinson described back in 1817 actually still holds true. There are a few things which have changed a little as time has gone by, but actually most of what he described is still how you would describe Parkinson's disease today. One of the most interesting things about James Parkinson that I've really found fascinating about him is that he was very much a Renaissance man. He was a physician and was trained as what we would now call a surgeon, actually, at the Royal London Hospital in the East End of London, but lived and worked most of his life in the village of Hoxton. And this is a map from around the turn of the 18th, 19th century, showing Hoxton as it was then, a little village surrounded by fields. For those of you who know Hoxton now, you'll appreciate that it's very, very different. It sits on the edge of the City of London and is a thriving and bustling part of the metropolis. Unfortunately, the house he lived in was demolished back at in the late 1800s. And this is the only surviving photo we have of the house that he lived in. If you go and visit Hoxton Square, there is actually a blue plaque that sits on the site of the house that he lived in. As I said, one of the things that is most interesting about James Parkinson is that he really was a man with many interests. In addition to being a physician and having written the pamphlet on the shaking palsy as well as a number of other medical pamphlets, he was also a geologist, and actually a very notable geologist. He was a member of the Royal Geological Society. He published a number of books, in particular about fossils and the way in which rocks form strata, and made a huge contribution to our early understanding of geology. He's one of the few individuals that I know of who has both a disease named after him, which obviously is very well known in terms of Parkinson's disease, but also a number of fossils. And this is an ammonite, an extinct form of mollusk, which is actually named after James Parkinson. This is Parkinson parkinsonii. If you go to the British Museum, his collection of fossils is actually on display there in one of the galleries. It was originally donated to the Natural History Museum, but is now on display in the British Museum. And there's a little explanation of his geological work in there. He was also a political agitator. So he wrote a number of seditious pamphlets. I suppose the equivalent these days would be tweeting. He would I was discussing this with a very eminent neurologist, and he said that actually these days James Parkinson would probably be a Corbynista. He was a radical, somebody who was interested in changing society. And again, a number of these, if you search for James Parkinson, he wrote under the pseudonym of Old Hubert. in his younger years, and was actually pulled up in front of Parliament. So he's associated with what turned out to be a fictitious plot to kill King George III, and was pulled up and questioned in front of Parliament because he wrote a pamphlet about it and wrote a letter about it. So he was very much an individual who was involved in lots and lots of different areas of life. But of course, most importantly, and his greatest contribution during his lifetime, was his essay on the shaking palsy. And as I said, I'd really recommend, if you're interested in Parkinson's disease, having a read through the pamphlet. It's relatively short and it's very easy to read. The key description in there that I've highlighted here is his original description of the disease that is now called Parkinson's. And he based this upon observation. So he was very much an individual who would look around him. This comes out in his work on fossils and geology. He observed the world around him and drew conclusions from that, a sort of very early observational scientist. And his grouping of symptoms into what we now call Parkinson's disease was based upon people that he saw walking around the streets of Hoxton and the streets of London, people who shared the same set of symptoms. And he described them in this way, as an involuntary tremulous motion with lessened muscular power, and with a propensity to bend the trunk forwards and to pass from walking to a running pace. It's already been highlighted that actually no two people have the same Parkinson's disease. But I think a lot of people who have an experience of Parkinson's would recognize one or other of those descriptions now as describing what they are experiencing of Parkinson's disease. He lived and died in and around Hoxton, and actually if you go to St Leonard's Church, which is just around the corner from Hoxton Square, this is an 18th century drawing of the church. If you go in there, there's a plaque that was erected in his honour many years after his death, describing his lifetime and his achievement. And I think it really is a mark of the respect and the debt that we owe to James Parkinson that we now name the disease after him. Which leads very nicely onto the next milestone that I'll talk about. And for this, we need to move 50 or 60 years into the future from 1817 into the 1870s. And this was a very tumultuous time in terms of neurology and the foundations of the field that we now know as neurology, resting on two cities, the city of Paris and the city of London, where groups of neurologists were working to characterize and really try and codify the different types of neurological symptoms and diseases that we see now. So one of the centres of this was the Hospital Salpêtrière, which is shown here, one of the major teaching hospitals in Paris. And a gentleman called Charcot, Jean-Martin Charcot, who is really one of the founders of what we now know as the field of neurology. And he carried out a huge number of clinics looking at patients, and there are actually a number of diseases which are named after Charcot, a number of disorders grouped together as Charcot-Marie-Tooth disease as an example of a disease named after Charcot. But he was instrumental in really taking the work that had been done by Parkinson and recognizing the disease that he described and bringing it together into a group of symptoms that could be used to actually diagnose people with the disorder. He grouped these together and talked about them in a series of lectures at the Hospital Salpêtrière on the diseases of the nervous system. This is a drawing from one of these lectures, which again sort of highlights one of the symptoms that James Parkinson noted, the propensity to bend the trunk forward. This is a patient that was seen in Charcot's clinics. It may not have been seen by Charcot himself, but it's one of the earliest photos of an individual with Parkinson's disease, where we actually know a little bit about the case history of this individual. Although we don't know his name, he was a metal worker who developed Parkinson's relatively early in life, in his 40s. And has coincidentally been used as a template for many of the images of Parkinson's disease as time goes by. This came about by the passage of this photograph from Paris to London, and another of the centres working on neurology at that time in the 1860s and 1870s. And a gentleman named William Gowers, who sits in the pantheon of neurologists with an equal level to Charcot. Again, William Gowers was very interested in trying to understand how neurological complaints present, the symptoms that they have, and how we can group together those symptoms to try and understand diseases and to try and develop therapies for them. And he wrote a vast treatise on the diseases of the nervous system that ran to 2 volumes, and wrote a chapter on Parkinson's disease. And this is really the era where we first begin to talk of a disease called Parkinson's disease. And both Gowers and Charcot recognized the debt that we owe to James Parkinson in bringing together those symptoms. In the diseases of the nervous system is this drawing, and the observant amongst you will notice that actually it's a drawing of this same gentleman from Paris. And this has been used in countless lectures and talks and published articles since that day to illustrate some of the features of Parkinson's. I won't run through this, dwell on this in too great a length. I think we've just had a really excellent description of some of the symptoms that we associate with Parkinson's. But actually the grouping together of those symptoms, the resting tremor, the freezing, the slowed movement, some of the psychiatric and cognitive impacts that Parkinson's can have, the grouping together of those symptoms as a cohesive disease entity really relies a lot on the work of Gowers and of Charcot. So we've moved from the original description of the disease through to really the definition of Parkinson's as a disease entity in the 1870s and 1880s. The next milestone I've picked upon is where we actually began to understand what was going wrong in the brains of individuals who developed Parkinson's. And there are a number of really quite major discoveries that our understanding of Parkinson's relies on in this area. But really, the first of these was back in the very early part of the 20th century by a researcher called Frederick Louis, who was a neurologist and neuropathologist who was interested in correlating what is seen in the symptoms of a disease, the neurological presentation of the disorder, with what was actually happening in the brain. And for this, what he did was he looked inside the brains of individuals who had died of problems related to Parkinson's. So if we were to look inside the brain of this individual from Paris from back in the 19th century, what we would be able to observe is loss of a region or damage to a region of the brain called the substantia nigra. In this cartoon here, it's the darkened region shown here and here. This sits right in the middle of the brain, in the region of the brain called the midbrain, also called part of the basal ganglia. And in the brains of individuals with Parkinson's, almost all of that dark coloration has disappeared. The reason it's disappeared is because the cells containing a chemical called neuromelanin, the same chemical that's found in our skin as melanin, those cells have all died. And as was pointed out earlier, by the time you start observing or developing symptoms in someone who develops Parkinson, 70 to 80% of those cells have disappeared. And that is visible to the naked eye. If you look in the midbrain of an individual with Parkinson's, you'll be able to see that loss of cells. And this was something that Louis picked up upon. He was looking in brain samples and able to observe that. But even more importantly, he noticed that with the cells that remained in these areas of the brain, there were great big lumps, amorphous lumps, which were found within those cells. He described them in quite some length in German. Unfortunately, my academic German is pretty rusty, so I haven't been able to read through his papers. But Louis was actually able to draw a series of pathological inclusions, he described them as, and we now know them as Lewy bodies. These are lumps of protein that sit within cells that remain in the brains of individuals with Parkinson's. And as we've learned more and more about these Lewy bodies, we've realized that actually these sticky protein lumps are critical to the development of the disease. And we really think of one of the aspects that lead to people developing Parkinson's disease. And I'll return to these Lewy bodies and the protein that we now know forms them in one of the other segments, one of the other milestones that I'll talk about. We've already heard a little bit about dopamine, and really the next major milestone, moving from the early part of the 20th century into the 1950s, was the realization that Parkinson's really rested upon, in terms of the symptoms, the loss of a particular chemical, the chemical dopamine. This came out of studies predominantly from a researcher in Sweden called Arvid Carlsson, who carried out a series of studies in rabbits, depleting their brains of dopamine. And those rabbits ended up developing symptoms that actually map quite closely onto what we see in people who have Parkinson's now, in terms of issues with their movement and their ability to move. What was realized by Arvid Carlsson was that this relied upon the fact that dopamine sits within your brain cells, within the neurons that form the networks that allow you to carry out movement tasks, as well as a whole host of other things. And that this dopamine acts as a neurotransmitter. It allows cells to talk to one another. When you lose the cells that contain this dopamine, the ability of those cells to pass on messages is reduced, and eventually, for people with Parkinson's, if enough of those cells are lost in the substantia nigra, then the ability to control and initiate movements is severely impacted. And essentially, at a chemical level, that is what causes Parkinson's disease, a loss of the chemical dopamine. This is a slightly complex circuit diagram showing actually where those brain cells are talking to each other. So this is the midbrain and the substantia nigra sitting here. And these red and blue lines represent bits of brain cells projecting out into other regions of the brain. In the normal situation, you have these projections going through a series of bits of the brain and eventually going out to the motor cortex and to your muscles and allowing you to control movement. What happens in Parkinson's is that those cells die off, and the signals, the control coming out from the midbrain is really drastically reduced. And as I say, at a fundamental level, this is what drives the symptoms that we now, or many of the symptoms that we recognize as Parkinson's. For those of you who like chemistry, there's going to be a little bit of chemistry chucked in here. And what was realized quite quickly after the discovery of, or the identification of dopamine as a neurotransmitter was that actually this might provide a route to providing some kind of symptomatic relief to those who have Parkinson's. And the vast majority of the drugs that we use today to treat Parkinson's disease come from trying to manipulate and modulate the dopamine levels in the brain. This is a diagram showing how dopamine is made. And actually, most of that isn't of great interest to us. The most important bit is the step that converts a chemical called L-DOPA into dopamine. Very early on, after Arvid Carlsson realized that dopamine was central to the control of movement, people thought, well, okay, if we have somebody who has Parkinson's, they have these symptoms. If we try and replace the dopamine that seems to be lost in their brains, perhaps we can make the symptoms better. And that was tried and unfortunately failed. And actually, we now understand why it failed. It failed because dopamine, if you take it as a pill or inject it into the bloodstream, doesn't cross the barrier between the brain and the rest of the body. It can't cross over into the brain. There's a very significant barrier between the brain and the rest of the body. Fortunately, very soon after that, it was realized that the direct precursor of dopamine, this chemical up here called L-DOPA, could cross into the brain if it was taken into the bloodstream or in the shape of a pill. Once it was in the brain, would be converted into dopamine and supplement the levels of dopamine in people who have Parkinson's. And actually, this is basically what the vast majority of drugs that we have for Parkinson's now do. Going back to that end bit of a brain cell, which is where it talks to the next cell and uses dopamine as a means of communicating. The drugs that we have either try and boost the levels of dopamine by supplying L-DOPA, increasing the levels of dopamine in the remaining cells, or they manipulate the breakdown of dopamine and prevent it being broken down so that the dopamine that is still there in the brain does its job a little bit better, a little bit more efficiently. We also have a group of drugs called dopamine agonists, which bind to the little receptors that dopamine activates and artificially supercharges them and increases the ability of those brain cells to communicate with the rest of the body. So that's basically where our understanding of the neurochemical basis of Parkinson's comes from. That's the basis for a lot of our understanding of the circuits that break down in the brains of individuals with Parkinson's. And what we've done is we've moved from the symptoms, understanding the symptoms, grouping them together, through to the major changes in the brain in terms of the degeneration and the accumulation of these Lewy bodies within the brain, to the chemical basis of the disease. Now that has helped greatly in terms of developing some therapies which are very, very useful for treating the symptoms, but it hasn't helped us in terms of developing therapies which slow down the disease or stop the progression in its steps. And this is really what we want to develop, something that will either slow down or even prevent Parkinson's altogether. And really, in the last 30 to 40 years, we've made huge steps in that direction of understanding the molecular basis, the actual biological basis of what causes those brain cells to die. One of the major advances was a story that could come out of a novel similar to the TV series Breaking Bad, about a chemist gone bad who becomes a drug kingpin in New Mexico. And the story actually goes back to the 1970s, and a student at the University of Maryland in America, a guy called Barry Kidston. Barry Kidston was basically a backstreet chemist. He was trying to synthesize a form of artificial heroin. This is morphine over here on the left. Barry Kidston was trying to make something called desmethylprotein, or MPPP. And he was trying to generate that to use as a drug of misuse. He was trying to use it, make it for his own use. When he was carrying out the synthesis reactions, and he was training as a chemist, so he knew quite a lot of what he was doing, he carried out one of the steps at the wrong temperature. And in addition to making this chemical here, which basically activates the same circuits that morphine does, he created a byproduct called 1-methyl-4-phenyl-1,2,3,6-tetrahydropyridine, which you'll be glad to hear I'll be referring to as MPTP for the rest of this talk. And that was a very, very grave error. MPTP is an exquisite neurotoxin. And when he injected himself with the MPPP contaminated with MPTP, what happened was that he developed the symptoms of Parkinson's over a very, very short period of time. He'd injected himself with a toxin which kills those very same cells in the substantia nigra that are linked to Parkinson's disease, but done this incredibly rapidly. And what we now know is that MPTP is converted into a chemical called MPP+, which is shown up here on the left-hand side of the screen. This gets into and is actually concentrated in the dopaminergic neurons of the substantia nigra, and then gets into the mitochondria in those cells. Basically, the mitochondria are the main energy providers of all of the cells in your body. If your mitochondria go wrong, you're in trouble. It causes huge problems. What happened in the case of Barry Kidston was that the MPP+, this toxin, was getting concentrated in the brain cells of the substantia nigra, was poisoning his mitochondria and preventing them making energy, and basically starving the cells that were in there and asphyxiating them, preventing them from functioning. That then caused them to die off and were no longer able to make dopamine, and eventually that resulted in him developing the symptoms of Parkinson's disease. What this highlighted was that the mitochondria, or mitochondrion to give it its singular name, is one of the critical components in the cascade of events that we think are leading to Parkinson's disease. So it's one of the key components. Dysfunction of your mitochondria is critical for many people who develop Parkinson's disease. Exactly how, we're not entirely sure, and there's a lot of research going into that at the moment. But the realization that these mitochondria were important go back to the original description of Barry Kidston's case. It was only realized that MPTP was so toxic when a group of drug users in San Francisco did exactly the same thing. They were provided with this MPPP, this artificial form of heroin, contaminated with MPTP, and then a number of individuals, I think just over 10, developed the same symptoms very early on in life. And it was realized through almost a detective story that what had caused their Parkinson's was exposure to this compound. So we're getting closer to the present day. That was back in the late 1970s, and that really the major step forward was the demonstration that if you gave MPTP to non-human primates, to monkeys, they developed exactly the same symptoms, providing a causative link between mitochondrial dysfunction and Parkinson's disease. The next and the final major milestone that I've talked about, and what I would highlight right now, is that I've not talked about a huge amount of research that's gone into Parkinson's. So I've really only taken snapshots of this journey that we've taken from 1817 to the present day. But for me, the next major advance was in 1997. And this was the discovery that alterations in your genes, and we heard a little bit about how some people who develop Parkinson's can inherit Parkinson's. It was the identification of the first gene for Parkinson's disease. And this goes back to looking at your DNA and looking at your chromosomes. In every single cell in your body, there is DNA which you've inherited from your mother and your father, which provides a template for how your cells function. Sometimes you get a version of this DNA which is incorrect. It's got a spelling mistake, if you like, that causes the proteins that our DNA makes to go wrong. And this was recognized actually for a number of years in Parkinson's, that there were cases where families you could see that multiple generations of a family were affected by Parkinson's disease. But the first realization, the first identification of an actual gene that was involved in Parkinson's didn't come around until 1997 and was based upon partly upon the work of Bob Nussbaum here, but as you can see, it was a large team who was working on this. And he identified in a family originally from the Mediterranean, from Greece, but had emigrated to America and was studying in a clinic over in America. He found a family where they had this inherited pattern for Parkinson's. This is the family tree published in the original paper. This is the most recent generation, the parents, grandparents, great-grandparents, and great-great-grandparents up here. And all of the squares and circles that have been filled in, these represent males and females in this family tree. All of the squares and circles that have been filled in this family tree developed Parkinson's disease. And as you can see, over half of the individuals in this family developed Parkinson's. As was noted at the start in the first talk, this is quite rare. It's estimated that somewhere between 1 in 10 and 1 in 20 people who have Parkinson's will have some kind of change in their DNA that might help cause them to develop Parkinson's. So this is a minority of people who develop Parkinson's disease, but in some cases we can follow it through a family like this. What Bob Nussbaum did is, working with clinical colleagues and geneticists, was to take DNA samples from the members of this family and to look through all of that DNA to try and find the individual gene which was changed, which had that spelling mistake, which was causing these individuals to develop Parkinson's. And he identified a mutation in a gene called SNCA. The protein is called alpha-synuclein. And almost immediately after this, it was realized by a group working in Cambridge, by Maria Spilantini up on the right-hand side, and Michel Godat, shown down here on the bottom left, that those sticky lumps that Frederick Louis had described back in the early part of the 20th century were made up almost entirely of alpha-synuclein. So here we have evidence from genetics, from studying families who have Parkinson's, that if you have a spelling mistake in the protein the protein alpha-synuclein, that they go on to develop Parkinson's disease. So this change in the gene causes them to develop Parkinson's. We then realized that actually, if you look in anybody who develops Parkinson's and has Lewy bodies in their brain, actually the same protein is found in there. So we think that something about the accumulation of this protein into these sticky lumps is giving us an insight into what's actually causing brain cells to die. And we think, and there's a huge amount of research that has gone into this, that something about the aggregation and forming of these sticky lumps causes brain cells to die in people who develop Parkinson's. We now actually have a much deeper understanding of the genetics of Parkinson's. We have a list of about 18 genes where if you have a mutation in them, it causes you to develop Parkinson's. We've also identified more subtle changes in the alphabet, if you like, which are much more common in the population. So many of us in this room will have one of these changes in our genes, but doesn't cause us to develop Parkinson's. It might increase the risk of us developing Parkinson's over our lifetime from the 1 in 500 people who have Parkinson's, as was mentioned earlier, increasing your risk maybe to 1 in 250 or 1 in 200, altering the risk based upon the genetics, your DNA that you've inherited. What's most important about all of these genetic discoveries is that they're providing us with the drug targets that was mentioned and that Parkinson's UK are trying to target. So a lot of the drug targets and a lot of the clinical trials which are ongoing now are targeting the genes that have come through studying families who have Parkinson's disease. So that's taken you on a whistle-stop tour of the last 200 years of Parkinson's research. I've missed out vast tranches of research that have greatly informed the way in which we understand Parkinson's. I haven't even touched on the cognitive and psychological side of Parkinson's, and hopefully you'll be hearing more about this in the coming hour or so. But I think what hopefully I've given you is a glimpse of the progress that we've made since 1817 and that initial recognition of something that we now know as Parkinson's disease, going through what that looks like in patients, how the brain has changed, the changes in the chemistry of the brain that come out of that, down to the molecular details of what actually causes those brain cells to die. And I think in a hopeful note at the end of all of that, I think that we really are beginning to move into an era where we will be developing drugs that make a difference. Over the next 5 to 10 years, those will be going into clinical trials. Some of them will fail. In fact, I suspect that most of them will fail. But as time goes by, we'll learn more about what works and what doesn't. And eventually, we will come up with therapies that slow down and perhaps even halt the progression of the disease. And I think, Tom will tell me if I've gone over, but I think that's about half an hour. So thank you very much for your attention.